Hello, my name is Margaret Harris, and I'm the industry editor at Physics World magazine. I'd like to welcome you to today's webinar on accelerating discovery with in situ electron microscopy, which is presented by Ray Unocic of Oak Ridge National Laboratory in the US and Jordan Mooring of Protochips. Ray is an R&D staff scientist at Oak Ridge's Center for Nanophase Materials Sciences in the Electron and Atom Probe Microscopy Research Group. He focuses on the development and application of advanced in situ scanning transmission electron microscopy, or STEM, techniques for nanoscience research. He also specializes in using aberration-corrected STEM in combination with other techniques to probe the atomic scale structure, chemistry, and defects of materials for structure property correlations. Jordan completed his PhD in material science and engineering, where his research focused on utilizing a range of techniques to characterize the deformation mechanics of nanocrystalline metals. He joined Protochips in 2016 and works with the marketing and product development team to explore opportunities for in situ tools and solutions. Jordan and Ray both welcome your questions, which you can submit any time during the webinar via the Questions tab on your screen. We'll have some time for them to answer questions at the end of the webinar, and also any unanswered questions we don't have time for will receive a response by email after the webinar is over. You can also rate the webinar at any point using the ratings button. Now I'll hand over to Jordan.
All right, well, thanks. Good morning, afternoon, and evening to everyone who's tuned in. Thanks for joining us. As she mentioned, my name is Jordan Mooring, and I'm at Protochips here. Today, I'll be going over some of the basics of in situ electron microscopy so that everyone can learn how these tools can be used to accelerate research discovery in their lab. So in my presentation, I hope to cover some of the basics of conventional and in situ transmission electron microscopy as it pertains to those tools' utility in scientific research. I'll then explain in more detail how our fusion heating and electrical biasing system works, as well as present a few examples from applications in 2D materials and memory devices. But I'll be keeping my presentation pretty brief today so that I leave plenty of time for Dr. Unocic to talk about some of the really exciting stuff his lab's doing. So you'll definitely want to stick around to hear more about their exciting work. So before I get into describing in situ techniques and going through our products, however, I want to give a really brief crash course on electron microscopy for those of you who may not be familiar with the technique. So an electron microscope is simply an imaging tool that uses electrons rather than photons to magnify and study specimens and samples. So in light microscopes, optical lenses magnify the image of the sample so that it can be directly viewed through the eyepiece. And in electron microscopes, magnetic lenses magnify the image of the sample so that it can be viewed on a fluorescent screen. By doing so, electron microscopes are able to resolve details that are much, much smaller than those for optical microscopes, imaging at magnifications over 1 million times. But there also are other benefits to using electron microscopes in addition to just tremendous resolving power. So when high energy electrons smash into atoms and matter, a wide variety of reactions occur, generating additional electrons, X-rays, and other signals that can be detected. For example, electron energy loss spectroscopy, or EELS, measures how much energy electrons have lost going through the sample, and this technique can be used to measure the electronic, thermal, and chemical states of the sample. Another technique, energy dispersive X-ray spectroscopy, or EDS, measures the characteristic X-rays that are produced from the electron beam to create a chemical map of the sample being imaged. Here you can see just a few types of the characterization capabilities that are accessible within the modern electron microscope. So because TEM is such a powerful characterization tool, it's been extensively used for research across the scientific discipline since the first microscopes became available many decades ago. So the first microscopes were relatively crude by today's standards, but you can see that over time their capabilities improved dramatically. So as the imaging resolution, analytical capabilities, detectors, and ease of use all improved, the TEM became a standard tool for cutting-edge materials research, especially at the nanoscale. But I actually want to draw your attention to the first in situ studies, which most people didn't know were conducted all the way back in the 1940s. But what does it actually mean when I'm saying in situ, though? Well, as I mentioned, TEM enables users to analyze their sample in a variety of ways. So from size and morphology to structure and chemistry, the microscope offers a comprehensive view into nanoscale materials. But this is really just a static view of a static sample in an inert environment. And the real world is much different from that. So with in situ techniques, samples are exposed to a stimuli or environment so that their dynamic behavior and evolution can be observed in real time. So this effectively turns the electron microscope from a simple observational tool into a truly nanoscale laboratory. Further, because sample behavior can be studied and captured over time, the creation of movies and animations from the data set can be created. So in essence, in situ techniques allow you to transform the one-dimensional characterization capabilities of your microscope into a rich multidimensional data set of the dynamic behavior of nanoscale materials and processes. So the capabilities of in situ techniques are quite tremendous, and they offer insight into never-before-seen reactions and processes in astounding details, simply seeing things that could not be seen before. But as I mentioned before, these techniques are not necessarily new. So the first studies were done back in the 1940s, but they were extremely limited in scope. Throughout the mid to late 20th century, in situ studies saw a slow decline as the reliability of the stages really wasn't sufficient. All of that changed over the past decade when MEMS-based sample control became commercially available. So the Protochips first system hit the market in 2009, and since then we've seen an explosion of research efforts utilizing in situ techniques, as you can see here from sort of the publication record. So previously, in situ techniques were viewed as tedious, specific, finicky, and delivered a blurry view of sample behavior at best. And this is where we're at now. So over the past decade, Protochips customers around the world have used our systems to publish hundreds of articles in top journals. This is a tiny fraction of all of the results that our customers have produced using our products, shown in some of these videos here. But I hope it gives you a quick glimpse into the tremendous possibilities afforded by in situ techniques. But so switching gears a little bit, I'd like to go into a little bit more detail about the in situ tools we'll be discussing today. So although we'll only be talking about fusion in detail, I always like mentioning that Protochips offers 3 in situ systems, which we've built to maximize the research possibilities available to any TEM lab. So this map just shows some of the major research areas we've seen folks utilize our tools. So from electrically cycling the liquid electrolyte in lithium-ion batteries to observing structural changes of nanoparticle catalysts in hot gaseous environments, there really just isn't much you can't do in the microscope these days regarding sort of in situ sample control. But today I'll be talking mostly about fusion. And as I mentioned earlier, it's our heating and electrical biasing system that allows you to characterize the thermal or electrical properties of your sample. But first, I want to talk in a little bit more detail about how the system actually works. So the MEMS-based E-chip is the core of the fusion system. And you can see here that a sample is placed directly onto the electron transparent silicon nitride supports that are fabricated directly into the E-chip. So the E-chip is then simply loaded into the fusion holder, where the electrical contacts automatically align with the chip, making a secure electrical connection every time. The holder is then loaded into the microscope just like any other holder, allowing your sample to be centered within the pole piece gap. I should also mention that we work really closely with all of the major microscope manufacturers, and that's to make sure that our products meet their rigorous standards for safety, compatibility, and reliability. So once the holder is loaded, you're basically ready to go. So what can you actually do with the fusion system? Well, one place we've seen a lot of interest is in the research and development of graphene. So as I'm sure you're all aware, graphene is a sort of wonder material that has the potential to revolutionize just about every facet of new technology. The problem with graphene is that it's really hard to make in large quantities that are also of high quality. And so there's a lot of research being done into the synthesis and control of graphene sheets. So some researchers from the University of Tsukuba in Japan were interested in the role individual atoms could play on the edge of the graphene sheets. So these are dopant atoms that could be left behind in the fabrication process or otherwise included in the graphene. And they also wanted to mimic the hot conditions of the graphene fabrication. So to do this, they needed to watch individual atoms moving around at high temperature while simultaneously ensuring that the heating membrane was not reacting with the sample so that the chemistry was preserved. And because we're talking about graphene, of course, they needed to do all of this at atomic resolution. And what you can see here are some of the results they found. So as you can see in the two movies, platinum atoms on the right, and the platinum atoms on the right move around and etch away the carbon atoms at the edge of the graphene sheets. And on the left, you can see an example of sort of copper and gold atoms, which had a different effect on the edge of the graphene. They actually mended the edge instead of removing it. So all of these studies showed how residual atoms left behind on graphene can affect the properties and the structure of graphene. And as you'll hear more later, the edge structure of these two-dimensional materials can have a profound effect on the properties and performance of the resulting sort of system. So this is just one example of 2D materials, and you'll hear a little bit more about other 2D materials that can be studied with fusion in a bit. But moving to a different application, I want to talk a little bit about memory devices. So recently there's been quite a bit of R&D into new technology called conductive bridging resistive access memory, or CBRAM. This technology uses 100 times less energy and is stable for far longer than other memory solutions that are available. But the way it operates is completely different. So as you can see in the top right, the on versus off state of the device occurs by the growth of a conductive filament between the two electrodes. So this filament growth isn't really well understood, and researchers at the University of Grenoble wanted to control, characterize, and observe this filament growth on a nanoscale device. So to do this, they needed very sensitive electrical measurements, while also being able to map the elemental composition and chemical migration across the device while it was being operated. To make it even more complicated, they needed to create a site-specific sample that they could attach to the E-chip contacts using focused ion beam lift-out techniques. So recently, we actually created a series of E-chips specifically designed to work seamlessly with common FIB techniques, letting anyone who's familiar with FIB attach their samples directly to our E-chips. So these FIB optimized E-chips, which you can see at the top, feature a notch design which improves electrical contact to the fusion system and actually allows final thinning of the sample to be conducted directly on the chip. In the top right, you can see a cross-section of the CBRAM device that the researchers were studying, and on the bottom you can see how the FIB sample is directly mounted onto the electrical E-chip. So once the sample was attached and thinned in the FIB microscope, they were able to electrically cycle the device and measure its electrical response in situ. And what they saw is up on the top left, which is the forming, reset, set, and additional reset phases. And this is the exact same response that they saw in bulk samples that were tested outside the microscope. So correlating the electrical characterization done in situ with ex situ devices was really important for them. But because they were in TEM, they were also able to characterize each of the layers before and after testing. And shown here on the bottom is just one example of a sample that they tested. And in this case, they deliberately overloaded the device to examine the chemical migration that occurs at device failure. So on the left is before device testing, and on the right is after failure. And the arrows indicate where the elemental redistribution and chemical migration caused a short across several layers, resulting in device failure. So wrapping up, I want to again mention that there's a lot of new science to be discovered using in situ techniques. And at Protochips here, we're doing everything we can to make our products in the fusion system powerful enough that they have the capabilities needed to conduct this groundbreaking research. So from chemically inert heating membranes and low leakage electrical contacts to workflows and technical support, the fusion system delivers in situ sample control that we feel can accelerate the research in any TEM lab. So with that, I'd like to thank all of you for your time and hand over the presentation to Dr. Unocic from Oak Ridge National Lab.
Okay, great. Thank you, Dr. Morin, for your presentation. It's great to hear all of the new developments and applications of in situ microscopy platforms that are readily available for scientific research. For those who may not know me or are just joining the webinar, my name is Ray Unocic. I'm a material scientist at the Center for Nanophase Material Science Division at Oak Ridge National Laboratory. And today my presentation is focused on the prospect of atomic scale edge engineering and 2D materials using in situ scanning transmission electron microscopy. Before I begin, of course, I would like to thank my collaborators on this work from Oak Ridge National Lab, Dr. Xiahan Song, Dr. Shufan Li, Dr. Christopher Lowe, David Gohegan, and Dr. Kai Shao, and also our theory collaborators from Ulsan National Institute of Science and Technology from South Korea. This is Wen Zhao, Jichen Dong, and led by Feng Ding. Okay, so within the past decade, we've had witnessed a tremendous growth in the field of materials research due to the development of new two-dimensional functional materials that are atomically thin. And when I say atomically thin, I mean just that. The thickness of these materials range from a single atomic layer to a few atomic layers thick. An example of these type of materials are graphene, transition metal dichalcogenides, moly disulfide or moly diselenide, and transitional metal dicarbides, et cetera, et cetera. So in the cartoon on the top right, you can see a wide variety of two-dimensional materials, and each of these materials have unique properties. So whether they can be insulating, metallic, semiconductor, or exhibit superconducting properties, it's really fascinating to see how a single class of materials can exhibit a wide range of unique properties compared to their bulk. And so these properties include electronic, optical, magnetic, and catalytic. And so the applications of these two-dimensional materials are for nanoelectronics, photovoltaic applications, energy storage conversion, water purification, and catalysis. So in materials research, our ultimate goal is to be able to understand and control the atomic structure and chemistry of these materials by either manipulating the atomic structure, for example, through doping, in this case a single nitrogen dopant in graphene, that can enhance the quantum capacitance of these materials, or the controlled formation of atomic-sized nanopores that, for example, can be used as separation membranes in water desalination technologies. So to understand the fundamental mechanisms of how we can actually synthesize and or tune the structure of these chemistries and materials, we turn to in situ electron microscopy. As discussed by Dr. Morin earlier, there's a wide range of scientific problems that can be explored using in situ electron microscopy techniques and platforms that are commercially available. One of the areas that we are investigating in Oak Ridge is how we can tailor the physiochemical properties of these materials by tuning the atomic edge structures, which I'll describe in great detail in the slides to come. We can use this to gain insight into thermolysis, which is the breakdown of molecular species to form new structures, and also use in situ microscopy to study the synthesis of new heterostructures, all at atomic resolution and all as a function of thermal exposure. So in order for us to attain atomic insight into these dynamic transformations, we need several essential pieces of scientific equipment. The first is an aberration-corrected scanning transmission electron microscope, that's shown here on the left, that is capable of resolving individual atoms, for example, in two-dimensional materials. Here at the Center for Nanophase Material Sciences, we have several of these advanced state-of-the-art microscopes. In this work, we use a neon ultrastem operating at 60 kV, which is below the knock-on damage threshold for most 2D materials, although it is recognized that the knock-on damage and ionization are inherent issues when imaging 2D materials, which I'll touch base on in a couple more slides. The second component is a MEMS-based in situ heating platform, such as the fusion system described by Protochips, in which we can transfer two-dimensional materials directly onto the heating chip platform and to perform heating experiments with controlled temperature at high accuracy, with high temperature uniformity, fast heating and cooling rates, and rapid stabilization with low drift. So a question that is often asked when performing these in situ heating experiments using these platforms is, how accurate is the temperature that you're applying? Well, if you're equipped with an electron microscope with an electron energy loss spectrometer, you can directly measure the temperature by measuring the energy shifts in the bulk plasmons, for example, as a function of temperature, in the work led by Matt Mecklenburg at USC and Chris Reagan at UCLA, published in Science 2015, or you can use optical phonons if you have a stem equipped with a monochromator for higher energy resolution EELS work that can actually pick up phonon vibrations. This is an example of some work led by a colleague of mine at Oak Ridge National Lab, Dr. Juan Carlos Drobo, where he was able to track the shift in the optical phonon signal as a function of applied temperature and hexagonal boron nitride, shown here left on the Z-contrast stem image. Now, on the right is the energy loss spectrum as a function of temperature, and the shift in the gain on the left-hand side and the low loss EELS signal is directly related to a combination of lattice thermal expansion and anharmonic phonon scattering. So how can we use this to actually measure temperature? Well, therefore, the local temperature can be measured purely from statistical principles, which relates to the probabilities of a transition from a lower to a higher energy state, and it's related to the Boltzmann factor. So simply by this equation and measuring the intensity ratio between the gain and loss peaks as a function of increasing temperature, we can determine how well we can determine the temperature accuracy in these microchip heating platforms with the nanoscale size probe. So as you can see here on the right-hand side graph, we're plotting intensity of the gain and loss peaks as a function of nominal applied temperature, and it fits very well with theory. So this gives us a great hope that we can use these in-situ heating platforms to study dynamic materials transformations, knowing that our temperature in our MEMS devices are accurate. So now that we have established control over the temperature in these devices, and now that we have an aberration-corrected stem that's capable of atomic resolution imaging, I'd like to focus the rest of my time now on how we can manipulate the structure of 2D transition metal dichalcogenides to alter their functional properties. So the atomic resolution image on the left is a typical stem image that we can acquire of tungsten-doped molybdenum diselenide, which has a hexagonal arrangement of moly atoms, shown in purple in the cartoons, and selenium atoms, which are shown in yellow in the cartoon as well. In the plan view and schematic views, you can see how the moly and selenium atoms are arranged in a hexagonal crystal structure. And if you look at the side view, we can see that the moly atom in central portion of it is bound to two selenium atoms. So in the stem, when we want to study the dynamic transformations, we can quite easily heat up these MEMS-based heating platforms and observe at atomic resolution what are dynamic transformations that take place. So this is the case where we take the two-dimensional material, heat it at 500 degrees C, and drastically from a nicely perfect sheet of tungsten-doped moly sulfide, we see dramatic changes in the structure. So quite evident are these big, large nanopores. So these are the black features in the image on the left-hand side that is decorated with these bright nanoparticles. And so these nanoparticles turn out to be molybdenum carbonide, as seen through the atomic resolution images of what the structure is and the EELS spectrum. So why am I saying that? Why is this important? Well, we're trying to study the dynamic properties of how edge structures form because we know they have important implications on functional applications. So we can actually use the microscope with the in-situ heating capabilities to transform them. So a question I often get is, what is the influence of the electron beam when you're doing these type of experiments? So this is a case where we're doing experiments at 500 degrees C, and we're watching the dynamic transformations happen. So with beam exposure, we do see the same type of formation of these nanostructures that we see without beam exposure. So the images on the lower right in panel A and B are locations that are far from the imaging regions that we were imaging during the course of the in-situ heating experiment, but they show the same type of structures. I'll get a little bit more in detail about beam effects a little bit later on in the talk. So I keep mentioning about edge structures and why they're so important. Well, it's been shown that if you have a two-dimensional material, the edge structures have enhanced properties. And so this is a case in the top left where you can influence the electronic properties simply by modifying the edge structure of graphene, whether it's in a zigzag configuration or an armchair configuration. And when you also do this, you can change the magnetic property as well. So this is a case where you're looking at modifying the zigzag configuration in narrow graphene nanoribbons, and it changes the magnetic properties and so on and so forth. In the materials that we're studying, these transition metal dichalcogenides, if you engineer the edge structure of these materials, you can significantly enhance the photoluminescence properties of these materials. So this is shown really nicely in the schematic from the paper led out of Penn State University on the lower right-hand side. So when we do these experiments, when we heat these 2D materials in the microscope, we see a lot of different interactions. So if you look at the lower panel of this slide, you see a lot of dynamic interactions. So these are actual experiments with the atomic resolution capabilities from our aberration-corrected stem, when we're heating it, we can see how atoms are actually rearranging and reconstructing. We can see how these nanoparticles are forming at these edges. And more importantly, we can see the formation of new structures inside the, starting from the initial 2D sheets of transition metal dichalcogenides. So we see that, that the transformation product is heavily influenced by the carbon residue that's on the surface. So when we transfer these materials directly onto the heating chip platform, we can locally tune the chemistry that way. And so people that do electron microscopy and they study transition metal dichalcogenides, boron nitride, or graphene, it's pretty ubiquitous that you'd have a lot of contamination on the surface. I like to turn that from a negative to a positive by saying if we have a little bit of carbon residue, or if we can intentionally modify the structure of these materials and heat it up, we can form some really interesting new nanowire or functional nanomaterials. So we notice that there's a dependence of these edge types on the chemical potential. And so when you're doing these experiments, you're in these materials, they're molybdenum and selenium atoms. We have the electron beam interacting with matter, and also the thermal exposure that's interacting with the substrate material itself. And so the lower energy of a selenium atom promotes the vacancies. So it's very easy to kick out selenium atoms from these structures simply by beam irradiation and simply by heating up with the electron beam. And it's a lot more difficult to move the molybdenum atoms. So if you look at the top panel right here, it's from, these are still images acquired from the dynamic stem image sequences that I've shown in the previous slide. If we have a heavy carbon residue, you can kind of see it looks a little bit blurry on top. And if we change that from a medium carbon residue to a low carbon residue, right away we can have dramatically different structures that's shown here in the lower left. And we think that if we can change the local chemical potential or the chemical potential of molybdenum, meaning that we can change the local chemistry on the surface of these 2D materials, we can alter the structure. And the way we do that, we want to be able to have some kind of parameter that we can tune. In this case, when we're looking at the transformation of molybdenum disulfide into functional nano edge structures, we want to be able to turn the knobs as far as temperature and as far as the chemical potential. And so we see here as we increase, if we increase the moly chemical potential due to heating, this increases the preferential formation of selenium vacancies during heating and beam irradiation. And so these excess carbon residue on the surface tends to change the moly chemical potential. And you can think about that as an affinity to form new structures. So if I have a lot more carbon residue on the surface, that I have change the local environment to a moly-rich, it tends to form these molybdenum-type carbide structures that we see in the lower left. But as you gradually remove a lot of the carbon residue from the surface, you can change the local chemical potential of moly, and then this is what's driving the formation of the new functional materials. And I'll get to these a little bit more detail, what the atomic structure actually means and how that relates to some kind of properties. But before I do that, I'd like to talk about a little bit about these edge structure nomenclatures. So you'll keep hearing in the next couple of slides when I say ZZ moly and ZZ selenium. So these are zigzag configurations. And if you follow my mouse, if these atomic columns, or these atoms and the moly atom in the purple, yellow, they form this kind of like the zigzag configuration. And the termination part of this moly. So this zigzag with the moly terminated edge, we call this a zigzag moly edge, or ZZMO. And then the selenium atoms that are terminated on this zigzag configuration, we, of course, denote the ZZSE, or zigzag selenium edges. We're forming new structures, as seen in the previous images. We tend to call these nanowires based on some fundamental work that was led here by Xiahan Lin a couple years ago when he was interested in shaping semiconducting molybdenum sulfide into metallic nanowires. So the edge structures that we see here that have that same type of atomic structure that's shown on the lower three images for the nanowires, we attribute that to formation of new structures that's similar to work that's been done by Dr. Xiahan Lin. So now that I gave you a little bit of flavor of how we can tune that structure and change the atomic structure simply by heating it, simply by changing the local chemical environment, whether it be a carbonaceous residue in a high or low concentration, we want to be smart about how we're doing this. So this is where we turn to our theory experts in an attempt to have some kind of control on a theory-guided synthesis of these edge structures. So our collaborators in Korea was able to take a wide variety of edge structures that are either zigzag selenium terminated or zigzag moly terminated edge structures, and calculate the formation energy required to transform into these edge termination groups. And so this is dependent on the local chemistry environment. So, for example, you have all these edge structures that's been predicted by theory. If we then calculate the enthalpy of formation of these edge structures as a function of the selenium chemical potential, the lines on this diagram basically shows which edge structure is more thermodynamically favorable to form. So, for example, if we're changing the chemical potential of selenium, so if I'm going more negative from negative 0.5 down to negative 0.1, this effectively creates a molybdenum-rich environment. So in that case, the enthalpy formation for example, forming these once a zigzag selenium terminated to moly-rich terminated configuration is highly favorable. We can also turn to our theory collaborators that can help us understand the reaction pathways that are important for understanding the atomic edge evolution dynamics. So here we're showing the atomic edge resolution images of a creation of a nanowire that's being formed in a region that has a low carbon concentration. So on the top right STEM video sequence, you can see the dynamic transformation that's happening. So if I point my cursor to this, you can start to see this nanowire that starts to form. All of these atoms in the molybdenum disulfide aluminum selenide sheet are atomically resolved. We can now see how each atom is rearranging at the atomic scale at elevated temperatures. So in this case, it's 500 degrees C. But more importantly, we can turn to our theory collaborators that can say how we can use density functional theory and how we can use ab initio molecular dynamic simulations to determine the energetic pathways that it would take to transform these molybdenum sulfide structures, and also to create these new structures based on the chemical environment. So they were able to do that, and in the lower right, we're showing how you can create a molybdenum terminated nanowire in a 30-degree orientation. Of course, the time scale in molecular dynamic simulations versus what we can do in situ microscopy experiments, there's a big difference. So we're talking on picoseconds for the MD simulations to seconds per frame for the STEM image simulations. However, the end result is roughly the same in that we're able to use the microscope to visualize the transformation products, and we can use theory to help guide and understand the transformation products that form from a thermodynamic point of view. So here's the bread and butter of my presentation. What I'm trying to convince the audience in the webinar today is how you can use two-dimensional materials, how you can tune the edge structure, and how we can use the electron microscope to transform one semiconductor material to something that has new and novel functional properties. So here we observe 4 stable edge configurations. So if you look at the top right-hand side, the top panel, these are experimental STEM images of the 4 most commonly observed edge structures that we have after we in situ heat this 2D transition metal dicalconide. We often do correlation with image simulations, so that this is the case of multi-slice image simulations to unambiguously determine what the atomic structure actually is. But it's clear from the STEM images, you know exactly where each atom is arranged, and you know based on the intensity that's proportional to the atomic number using this the so-called Z-contrast imaging technique, roughly what atom is present in that case. But more importantly, we can take now from the experimental observations of these 4 thermodynamically favorable edges, we can create a structure model, and then we can determine through Ference principles calculations what exactly the properties you have at the end of the day. So I mentioned earlier that molybdenum selenide is a semiconductor, but you can actually change that structure completely if you were to engineer the edge structure simply by heating it inside the microscope. So in this case, we're plotting the density of states. And so for this configuration that's a zigzag selenium terminated with a little nanowire at the end, it changes from a semiconductor to ferromagnetic. We have the density of states that show the spin up and spin down configurations for that specific structure that's forming at the edge. We have a case that we're changing from a semiconducting material for purely metallic, and then also a specific configuration that we can see that it's a selenium atom terminated and it still retains a conducting nature. So why am I showing this? Why am I trying to go to a lot of detail of how we can rearrange atoms? Well, like I said earlier, if we know the structure and chemistry, we can change, and we have the ability to tune it, we can change the properties significantly. So this example basically shows how we can take a material, transform it in the microscope, and obtain new structures and relate that to new functional applications. So hopefully one of these days in the near future, if there's a technological need where you have a two-dimensional material for a nanoelectronic application, for example, and you want to have a thin metallic region, or if you want to have a ferromagnetic region that's locally confined, maybe this is a way to do that. So in conclusion, I hope that I was able to convince you that in situ STEM provides atomic resolution insight into the dynamic edge reconstruction process, and we use this to study new functional properties of the 2D transition metal dichalcogenides. Of course, there's a lot of research not only here at Oak Ridge, but worldwide of how you can use these platforms to understand the dynamic transformations all at atomic scale. But in this specific study, I was hopefully able to convince you that if you change the edge structure that's formed by simply changing the local chemical environment, you can create new thermodynamically stable and metastable structures in situ that can be predictive quite well with DFT and ab initio MD simulations. And the most exciting thing at the end of the day with this type of research is what new insight that can be gained of how the folks that do synthesis and device fabrication, how you can use this knowledge to synthesize new functional materials with pores that are decorated with nanowire edges and form new novel 1D to 2D nano heterostructures that are either semiconducting or metallic or magnetic. So I'd like to end my slide saying that I'm a scientist at the Center for Nanophase Material Sciences. It's a Department of Energy National Science user facility that's supported by the Department of Energy to work with people in the United States and worldwide that has access to use the instruments and facilities here at Oak Ridge to do this type of research. So the research areas that we focus on that we're experts on are synthesis, nanofabrication, advanced microscopy, like I've shown you in this presentation today, functional characterization of materials, and also theory and modeling. So this is a user facility that anybody can write a simple 2-page proposal and have access to the unique instruments and have access to staff expertise and perform experiments like you've seen today in this presentation. So with that, I would like to end my technical part of the talk, and I'll hand this over to Dr. Mooring for the question and answer sessions.
Okay, well, thank you both, Ray and Jordan, and we'll start taking some questions from the audience. Please do go ahead and continue to write in your questions. You can ask questions either of Jordan or of Ray. So the first question is, let's see, how are your products safe to use in a microscope? I think that one's for Jordan.
Yeah, we get this question quite a bit, especially with our liquid and gas cell. It can be a little nerve-wracking when people think that, about putting liquids in the microscope. But yes, they are. We work with Thermo Fisher, we work with Neon, we work with Joel. They actually have quite a bit of quality control that they do on our products before we were certified to put them into the microscopes. So it took several years, and it's a continuing thing that we work with them to make sure that our products work in the microscopes. If you have a different microscope or a particular old one or a particularly new one, you're always welcome to reach out to us to find out about the compatibility. But for the most part, if you have a transmission electron microscope, not only are our products safe to use in that microscope, but they've also been certified by the manufacturer of that microscope to be safe.
Okay, so next question, I think it's also for Jordan. How do I know which E-chip I should use for my study?
Yeah, so Protochips, our background as a company actually is in MEMS fabrication. So that's, our founders have background both in the R&D side as well as the manufacturing and quality control side of MEMS fabrication. And what that has allowed us to do is create quite a wide array of different E-chips depending on the type of experiment that you're looking to do. So when someone buys a system of ours, we have an ordering guide that goes through all the different electrical configurations, different electric contacts, different membrane supports, and we actively work with you to sort of help you pick the best chip for your application, but we also have some general guidelines for that. So it would depend on your application, but we do offer, I think, over 30 different combinations of chips to try to cover the whole realm of science.
Okay. Next question here is again for Jordan. Please believe me, you can go ahead and ask great questions. We're happy to have questions for both presenters. So someone has asked, has anyone used the fusion holder to simultaneously heat to a high temperature and also apply bias at the same time?
Yes. So I didn't cover that today, but the fusion system does enable electrothermal measurements. So just offhand, we've had some customers of ours publish work in the research areas of field-assisted sintering. So that's heating ceramic particles up to high temperature and then seeing how they agglomerate as a function of electrical field. We've had some customers look at ferroelectrics and piezoelectric materials. Those are temperatures that have phase transformations that are correlated with electric field and high temperature. We've had a few individuals look at memory devices or sort of nanowire type applications. So there's quite a few, and I think on my slides I had a link at protochips.com/bibliographies. That's a comprehensive database of all of the publications that our customers of ours have produced. So I encourage you guys to go to that site and look through our publication record, or our customers' publication record for yourself. But yes, plenty of people have done that.
Okay. Another question just come in from someone who says he works on polymeric nanofibers, and the question is, how do you load such a delicate sample onto a very small holder? Yeah, I guess I can answer that one as well. So sample prep is, I think as anyone who's used an electron microscope or TEM knows, sample prep can be one of the more difficult ways of doing it. We would work with you to try to figure out how you could put those nanofibers on. There's a lot of drop casting methods that are possible, ex situ techniques, in situ, fib lift outs. There's quite a few different ways of doing it depending on the sample geometry and chemistry. But yeah, to answer the question in more detail, I think I need to know more about the fibers. Okay. Another question, are there any shared technologies to transfer 2D materials to Protochips MEMS chip? Someone says that they're having a hard time to transfer their 2D material, which is graphene and molybdenum disulfide, to the heating chip.
I guess that could be a great question for me. So if you read the paper that we published in Nature Communication, it describes quite nicely in the method section. But basically, when you grow either with CVD processes on silicon or silicon dioxide wafers, you can etch away you can first spin coat some polymethyl methacrylate on the surface of your 2D material, dissolve away the substrate, and then transfer using a wet transfer process onto the chips themselves. So in this case, we use a little funnel, and then we basically evaporate the liquid that's the 2D material floated on until it is positioned right on the microchip device. And then, of course, cleanliness is an issue. We have cases then we take the entire microchip, dissolve it in, immerse it in acetone and methanol to remove some of the residual PMMA that's on the surface of those microchip devices. So I'd be happy to share any more if you send me a private email. The other thing that I do want to mention is when you do the transfer process, and to be able to see at atomic resolution, so the fusion heating chip microchip devices can either have a silicon nitride membrane, can either have a carbon membrane that's a support material. We like to use the silicon nitride membranes that we modify with the focus ion beam instrument to create an array of tiny holes through which that we can see at atomic resolution. So it's basically you're putting your 2D material on the chip that's supported by silicon nitride, but still has holes in it so you can see at atomic resolution.
Okay. Another question for Ray, this time about some of the fantastic videos you showed in the presentation. Someone wants to know what time resolutions were the videos of atom diffusion taken at?
That's a great question. So inherently, STEM imaging, since you're taking a finely focused STEM probe that's atomic size and you're scanning it through the material and you're rastering it, basically it's a little bit slow process. We don't have really fast dynamics. We're in something we're doing in the microseconds per pixel time resolution. So per frame, to get atomic resolution that you can easily see all the atoms can either be between 1 and 4 seconds per frame. So it's a little slow process, but again, there's a trade-off between the spatial resolution that you want to get and the temporal resolution that you hope to be able to see some dynamic transformation. So there's a good trade-off between spatial and temporal resolution, but it is a little bit inherently slow as compared to other TEM techniques, just for example, TEM imaging using fast detectors or dynamic TEM.
Okay, another question is about characterization. And so someone asked, is my characterization affected by tilting the sample?
So the great question.
Yeah.
Sorry, go ahead.
Do you mean to answer that, Ray, or do you got it?
Yeah, go for it.
Yeah. So the tilt ranges of the holder, can somewhat depend on the microscope. So the microscope that Ray is using is a neon instrument, which is quite a bit different from a conventional STEM. But in general, we build the fusion system to have double tilt functionality, so that's tilting in alpha and in beta. And the electrical contacts remain in contact with the chip at all times. So as you're tilting, the temperature stability is maintained, the electrical stability is maintained, and so all of the electrical connections tilt with your sample. So no, your characterization capability isn't affected while tilting if that's a type of analysis that you'd like to do.
Okay, got a couple more questions for Ray now. One of them is, are you aware of any in situ electrical biasing studies of 2D materials at atomic resolution?
I'm sure there is. There's some excellent work done at the UK and in Japan that are pretty interested in performing biasing studies to look at, for example, in graphene, how you can form 5-7 stone wheels type defects simply by putting electrical bias on it. So there are a lot of work out there. You just have to search the literature for that.
Yeah, and some of the electrical studies that I showed, I went through pretty quickly, but especially recently and in some publications using the FIB optimized chips, we've seen atomic resolution under different fields and different current conditions.
Okay, we got a follow-up question to Ray's answer about time resolution. Have you tried imaging the atom diffusion along 2D material edges using transmission electron microscopy rather than STEM?
That's a really good question. So we have done that. The second part of our research is we wanted to use amorphous precursors and study how these materials form layer by layer using a technique called thermolysis, or breakdown of these molecular species. We performed experiments both on the STEM at atomic resolution and near atomic resolution using HRTEM and our FEI or Thermo Fisher Titan microscope that's equipped with a one-view camera. So we've tried that to increase the temporal resolution a bit. So absolutely, we have done that.
Okay, another question for Ray, asking whether you could see single dopant atoms stabilize within pores in the monolayers of 2D materials.
That's an interesting question. I think by definition, if you have a pore, there's nothing there, so it's really hard to stabilize. However, when we do some of these experiments, we have a thin layer of amorphous material that we can sometimes see single atoms position in. So, for example, if we're etching away the material, and we create this little nanopore, there's a really thin layer of amorphous junk, I guess, left behind that we can see single atoms on. But if you don't have anything to support it, it's very difficult to have a dopant in there.
So amorphous junk is my new favorite technical term. Okay.
Yeah.
So now, quite general question, I think for Jordan. How do you know the temperature of the sample?
Yeah, so Ray actually went over some of that in his presentation. There's a lot of different ways to look at the temperature. All of our E-chips are calibrated with the optical pyrometer, so it's a sort of thermal map of each chip before it gets shipped out. And those chips that optical pyrometry is used to create a unique calibration file for each chip. So I don't know if you want to talk any more detail about the sort of correlation that you guys derived, Ray, with those temperature measurements. But yeah, that's the general approach.
Right, and so that's something that we wanted to test, and as scientists, we want to really understand the type of measurements that we're doing. So for giving, say, for example, microchip heating devices that has some kind of calibration from cameras that Jordan just mentioned, we want to be able to confirm them for ourselves. So that's why my colleague Juan Carlos Idrobo took an initiative to be able to understand temperature measurements at the nanoscale using a nanoscale probe using EELS measurements. So we've shown that in I can't remember the slide number, but we have that documented in a PRL paper. And of course, the group at USC and UCLA that use bulk plasmas to measure shifts in the energy and directly relate that to temperature. So there's ways to independently verify what temperature you're at simply by doing electron energy loss spectroscopy measurements.
Okay. A question is, when you're running the in situ experiments, do you need the special software, a special suite of software to run those experiments?
Yeah, I didn't mention this in my presentation, but all of the fusion systems come with a software that we designed. We call it Clarity. The Clarity software runs the fusion holder, and so that has different workflows and drop-downs to sort of help guide you through the uploading of a calibration file, for example, and then running an experiment itself. So the Clarity software comes with Fusion, and if anyone has any questions about that software or just in general how to run in situ experiments, feel free to email me. I think I left my email on my first slide, but I'll also, but you just send me an email if you have questions about software running experiments.
Okay, we still have a bit more time for questions, so if you do have another question, please do go ahead and send it in. We have had one question about whether people would be willing to provide their PowerPoint slides. I'm not sure. I'll give that to Ray and Jordan for their specific slides, but if you'd like to watch the webinar again, it will soon be available on the Physics World website. So you'll be able to find it there, and please do go ahead and recommend it to your colleagues if you've enjoyed it today. Okay, time for a few more questions. Let's see. Someone points out that from the still life images of TEM 9 years ago when this person retired, they're seeing enormous leaps and bounds. If you're rearranging atoms to manufacture a wire structure, how do you manufacture on a larger level?
Jordan, maybe I can fill that question. So there's been a big initiative of how we can use electron beams to manipulate matter down to the fundamental length scale or the atomic structure. And so this is definitely not a mass production type process, but more using the electron beam to understand the basic mechanisms of how and why these materials rearrange themselves from a basic level. But of course, a lot of people want to be able to mass produce this if you wanted to create functional, these 1D and 2D heterostructures. So one way you can do that is using larger scale, like a factory of like electron beams, and maybe even multiple electron beams to do that. But in the example that I showed you, it looks like if you take these materials that's not radiated by the electron beam and thermally expose these in vacuum at elevated temperatures, you can create the same type of edge structures that you've seen here. So that could be a nice way to mass produce these type of materials. And then once you've done that, then you can do the standard transfer processes to actually create these nano structure devices from their substrate.
Okay. Well, thank you to everyone in the audience who asked a question. It's always great when audience members are so engaged, and this presentation really seems to have hit a nerve with some people. Lots of really interesting techniques on display here. So thank you to everyone in the audience who did ask a question. If you think of a question later, or you can contact the presenters. They did post both their email addresses, I believe. And as I said before, if you'd like to watch the webinar again, it will soon be available on the Physics World website. Thank you again to Ray and Jordan, and thank you to everyone who's joined us today. This webinar is now concluded. Thank you very much for attending. Bye-bye.